you're supposed to one side. So I'm trying to manage as the case may be. So you have Golgi body. Golgi what? Body. You see it now. Then there's another structure again. Very tiny structure like this. That structure, maybe I will label it. Let me try and see if I can get it here. This structure is called lysosome. Lysosome. Okay? This lysosome is a spherical, is a tiny granules found in the cytoplasm. This lysosome is responsible for secreting what we call lytic enzymes or digestive enzymes, as the case may be, but lytic enzymes basically. So this lysosome helps to prevent and fight against foreign body, but it prevents the cell from carrying out what we call autophagocytosis. I write it out. So lysosome is responsible for preventing what? Autophagocytosis. For those of you writing, I will spell it for you. Auto, A-U-T-O, phago, P-H-A-G-O-C-Y-T-O, autophagocytosis, S-I-S. What does it mean? It simply means cell eating up itself, cell destroying itself. So this lysosome prevents the cell from destroying itself. Now, you know, I made mention of something about the Golgi body. I told you that it is a saucer-shaped membranous structure that is involved in packaging. And this structure, this is where glycosylation takes place. I told you that's the Golgi body. And what is glycosylation? Glycosylation is the process that involves the conjugation of glucose and protein or glucose and lipid. So when glucose and protein or glucose and lipid is combined, joined together or conjugated, that process is called what? Glycosylation. Let me spell it for you. Glycosylation. G-L-Y-C-O-S-I-L-A-T-I-O-N. Glyco what? Silation. Is that clear? Then there's another structure again I need to show you. That structure, we call it centrome. It is basically found in animal cell. When I'm going to draw the plant cell, I'm just going to draw the cell and we'll just analyze. Now, this thing is found at right angle. Okay? It's a rod-shaped structure found at right angle. That structure is called centrome. Just take it here. Centrome. See it here. Centrome. C-E-N-T-R-O-M-E. So the centrome is responsible for cell division. It's basically found in plants. There's another structure again I've not shown you. That structure is also somewhat spherical, and that structure is found in cytoplasm. Let me label it this side. Okay, let me take it down. I have the space there. It's called vacuole. Vacuole. Okay, the vacuole is an enclosure that is found in the cytoplasm. An enclosure found in the cytoplasm. In plant, the vacuole contains what we call cell sap, and the cell sap contains sugar, salt, and some pigments dissolved in water. Now, in plants, this vacuole can sometimes become turgid, okay? Become turgid, and when it's turgid, it can give protection or mechanical support to herbaceous plants. Do you get it now? So, with this, we've been able to analyze the various components of the cell, especially for animal. Even these similar things with plants. I think the only difference is that there's no chloroplast, as the case may be. We don't have that. And then in the case of plants, there is a large vacuole. The vacuole of plant is larger as compared to that of plants. Do you understand what I'm talking about now? So in our next video, I'm going to draw the plant cell, and then just to do a recap of what has happened. So let me give you a recap because this video, I'm going to break it down. I'm going to divide it because it's about 24 minutes. So I will split it into two so that it'll be easy for you to be able to, I don't want it to be too long. Don't forget the recap is we have Golgi body. I told you it's a packing membranous structure. It's saucer shaped. Glycosylation takes place here. I've told you what glycosylation is. Conjugation of glucose and protein, glucose and lipid as the case may be. And I also told you we have what we call ribosome. Ribosome is the site where protein is synthesized. I told you mitochondria, a rod-shaped organelle responsible for producing energy. That's the powerhouse of the cell. Contain cristae and matrix. Okay, I also told you the cytoplasm. I've explained that in the previous video. And I told you the nucleus. In the nucleus, even in the nucleus, you have what we call nucleolus. Nucleolus simply means little nucleus. And in the nucleolus, they are responsible for forming some parts of the ribosome by producing what we call the rRNA. That small R means ribosomal ribonucleic acid. And then we also have what we call the chromatin granules, which is also known as the chromosomes. And I told you that the chromosome become visible during cell division and is at the prophase stage that you have that. This chromosome houses what we call the DNA or the gene, which is the genetic material that controls the thread that carries the feature of the cell. Okay, so the DNA is known as deoxyribonucleic acid. Let me write it for the sake of people who are writing. So let me write it here. Just see it. Deoxyribonucleic acid. So in some text, they will tell you that the nucleus contains nucleic acid. They contain nucleic acid. Oh, that reminds me, they also have what we call nucleotide. They also have what we call nucleotide. Okay. This nucleotide, they are inside the nucleus. They are newly synthesized ribosome. Okay. Release into the cytoplasm. This nucleotide, they are newly synthesized ribosome released into the cytoplasm. So this work is coming directly from the where? From the nucleolus. You know, I told you, ribosomal ribonucleic acid. It makes some part of the ribosome. So nucleotide, they are newly produced what? Ribosome that are released in the cytoplasm. They are also found inside of the nucleus. So the nucleus is like what is housing all of them. So when you are talking about nucleus, you make mention of what we call the chromatin granules or the chromosomes. You make mention of what we call the nucleolus. You also make mention of what we call the nuclear membrane that regulates exchange of material between the nucleus and the cytoplasm. Also, there's what we call the nucleoplasm. Okay, this is a kind of gelatinous fluid inside the nucleus. Just take it that way. So we have nucleoplasm, we have nucleolus, nuclear membrane, and the chromatin material. Listen to the video over and over so that you can digest it and get it clearly. Then we now also talk about the lysosome. The lysosome secretes lytic enzymes and it prevents the cell from carrying out what we call autophagocytosis and also fight against foreign bodies in the cell. We also made mention of centromes. Okay, centrome, C-E-N-T-R-O-M-E. It is also a structure that is found at right angle by the side of the nuclear membrane. Its basic function is for cell division. That is what it does. Then we also looked at ribosome. It's responsible for the synthesis of protein. And when you find ribosome on the endoplasmic reticulum, you call that endoplasmic reticulum, which is a channel that transverses or passes through the nuclear membrane into the cytoplasm. So that endoplasmic reticulum that contains ribosome is called rough endoplasmic reticulum because of the presence of ribosome that synthesize protein around that region. But the smooth endoplasmic reticulum are deficient of ribosome, and so they are responsible for transporting corticosteroids, steroids and lipids. And then you want to move on, we also talked about the vacuole, which is almost insignificant in an animal cell. So this is an animal cell, that's why it's not too big. You understand? So the vacuole is an enclosure found in the cytoplasm of the cell, as the case may be. It's there to temporarily store some things. It contains cell sap, sugar, salt, and pigment dissolved in water in the case of plants. It gives plant mechanical support when it is turgid, especially herbaceous plant. And then we also went on, let me show you, the outer layer is called what? The plasma membrane. Okay, so let me just put it here. This side is called the cell membrane. That's the outer layer of this. Okay, you see the reason why explaining it will help to remember. Cell membrane, or we call it plasma membrane. Okay, I have already explained what all these things are in the previous video. So this is cell membrane or plasma membrane, as the case may be. Now around this side, let me just add for those of you, because of A level and those people too who might be writing JAMB as the case may be. Around this side, there's something we call micropinocytes. Let me just write it. In plants, in the case of plants, we call it plasmodesmata, but in animal, we call it micropinocytes. Pinocytes. Now, what is micropinocytes? These are tiny pores, materials found outside around this plasma membrane. They help to exchange of materials to take place between the cell and the outer environment. Okay, micropinocytes, where something like pinocytosis will take place and phagocytotic processes will take place. So when you talk about pinocytosis, it has to do with cell taking in water. Pinocytosis, cell drinking, cell taking in water. Phagocytosis, cell taking in food substance or substances as the case may be. I want to wrap this class up. I'm sorry that this class, each section might be 15 minutes. It's biology. Biology is very broad. Don't be angry. If you're not following me, please, I beg you to follow me. I will divide this video into two. The first part of this video will take some part, and the second part, 30 minutes is too long. So thank you. I'd like you to follow and share the video if it's possible to help your other persons who need to come up in the subject. Thank you. See you in the next video.